Okay, so I hope everybody is now online. Good evening, ladies and gentlemen. My name is Kristina Beblo-Vranesevic. I'm from the DLR. And in the next 30 minutes, I will tell you something about the microbial life in extreme and dry environments on Earth, and maybe also a little bit about the life beyond. So first, some words about the DLR, the German Aerospace Center. DLR is divided in 3 parts. We have the Research Center for Aeronautics and Space of Germany. We have different research and development activities in the fields of aeronautics, space, energy, transport, security, and digitalization. The second branch is the German Space Agency at DLR, and the German Space Agency plans and implements the National Space Program of Germany on behalf of the government. And the third part is the project management, and this branch is overseeing funding programs and supports us in the knowledge transfer. DLR has some institutes and facilities all over Germany. So we have 30 locations and about 10,000 employees at DLR. The headquarters is in Cologne, and we have also some offices in Brussels, Paris, Tokyo, and Washington. We are sitting in the facility or in the area of Cologne. I'm located in the Institute of Aerospace Medicine at the Department of Radiation Biology, and I'm in the Astrobiology working group with the head of Petra Redberg. Our department is divided into 2 buildings. So we have this older building and this newer building. And in both buildings, we have microbiology labs, and we have also in our basement the space simulation facilities, which is a very unique working environment at DLR. But now I want to get more into the real topic of microorganisms, and will tell you some basic facts about microorganisms on Earth. In general, we have 10 times more microorganisms on our human bodies than human cells. So we have 1 × 10⁻14 microorganisms on or in our body. In only 1 gram of soil, we can find about 1 × 10⁹ microorganisms. And if we sum this up, a really big number is coming up, and we have 10 million more microorganisms in our oceans, and then stars in our universe. And if you see this, the total bacterial and archaeal cell number on Earth is about 1 × 10⁻13, which is a really unbelievable big number. And one thing which is very important and to keep in mind, microbes are everywhere. They are in the sea, they are in the on the bottom of the ocean, they are in the soil, in the deep surface, on all organisms living on the land, and even in the air. But today I want to talk more about extreme environments on Earth. First, I will give you a short definition. What is extreme? In general, the term extreme is a very anthropogenic point of view, because we as humans think something is extreme which is not living under our conditions. And these extreme conditions can be either chemical or physical. So for example, the pH value, the salt concentration, the redox potential. We can have different high or low heavy metal concentrations, and the physical extreme conditions can be in the range of temperature, pressure, radiation, or also water availability. However, under these extreme conditions, we find microorganisms and also small eukaryotic organisms, which are able to live in these or under these extreme conditions and in these extreme environments. And these microorganisms are called extremophiles. And which is very important, and also you should keep this in mind, extremophilic organisms not just tolerate these conditions, they really need these conditions. Under normal conditions, which are normal for us humans, they cannot survive or live. And what extreme environments on Earth are existing? So in general, also here we have very diverse environments. They can be very cold in permafrost or polar regions. They can be on the bottom of the sea. They can be on land, some terrestrial extreme environments. We have deserts, but there are also some artificial extreme environments, for example, nearby nuclear power plants. And in all these extreme conditions, on land, on sea, we find microorganisms living there. We have the psychrophiles, which are cold-loving organisms. We have hypothermophiles. We will hear something about them in some seminars later this year. We have xerophiles, acidophiles living at low pHs. We have the piezophiles. Petra Schwentner already told you about them. And we have the halophiles. Today, I'm focusing on the organisms living in dry areas. And here are some more information about the organisms I will talk today about. I will talk about prokaryotic microorganisms, bacteria, and archaea. In general, the size of these cells is about 1 to 2 micrometers, and the cell is the smallest building block of life and takes over all functions of life. So we have one cell which is doing all metabolism for its own life. And within this cell, we have, of course, we have the DNA, we have maybe some plasmids, there are ribosomes, we have some cell appendages. In between, we have the cytoplasm and we have the cell wall, which can be composed by several components. But what is happening if the cells get dried or if they are confronted to desiccation? All the cellular bound water, which is in the cell, for example, here in the cytoplasm, also on proteins and also on the DNA, leaves the cell, and this loss of water is causing several damages. And just to make it short, all cellular components are affected. For example, in the DNA, we get single and double strand breaks, we get protein DNA crosslinks, the conformation of the DNA itself change from the B conformation to the A conformation. We have in the proteins, which are in the cytoplasm, protein denaturations and perturbations. The cell wall or the lipid layer is affected. We get, for example, spherical micelles. And of course, also due to the desiccation stress, we get oxidative stress and some reactive oxygen species are produced. And these reactive oxygen species can affect, secondly, again, the different compartments in the cell. So if you take this into account, we see that the damages of ionizing radiation, for example, the production of reactive oxygen species and single and double strand breaks, are very similar to the damages of desiccation. And the same is somehow true for osmotic stress, because if cells are under osmotic stress, they are also confronted with desiccation. So one point was that we thought, okay, maybe protection mechanisms which are active during osmotic stress and repair mechanisms which are active after exposure to ionizing radiation can help the cells also during desiccation. Here are two pictures of one natural and one artificial area where we have higher levels of radiation, some sand in Brazil and nearby a nuclear power plant. And even in these areas, we find microorganisms who can survive or even live there. And maybe the one or the model organism in radiation biology is Deinococcus radiodurans. We see here a scanning electron microscopic picture of the cells, and here is Deinococcus growing on a plate. And Deinococcus is one of the record holder concerning ionizing radiation. The D10 value of Deinococcus is 12.7 kilograys. And just to compare, the D10 value for E. coli, another mesophilic model organism in microbiology, is 250 grays. And just to compare with our sensitive species of humans, our D10 value is even at 10 grays. And the question was, can protection mechanisms against radiation stress also protect the cells against desiccation stress? And which protection mechanisms are active, for example, in some microorganisms? The first one is polyploidity. That means that in the cell, not only one copy of DNA, you can find not only one copy of the DNA, there are several copies, up to 40 copies of the same DNA. The DNA itself has a toroidal, very winded structure. And we have a special manganese and iron level. We have a cell membrane with pigments. You already saw this in the picture of Deinococcus, where you have these pinkish colonies. And these cell membranes are helping during UV radiation, and also again are helping to fight against this reactive oxygen species. In these cells, we have efficient DNA repair mechanisms. And as you already saw in the scanning electron microscopy picture, we have D and tetracocci. That means that the cells are sticking together by 2 or even by 4. This is all true for Deinococcus radiodurans, the record holder. But there are all other radiation-tolerant microorganisms which do not possess these adaptations, and they are also desiccation tolerant. And I will give you some examples now from our lab. We tested 3 different organisms. I will explain these organisms to you some slides later. So we have Butteoxella, Salinisphaera, and Yersinia, and we dried them up to 190 days, and we saw that the survival rate is going down. We also irradiated them with ionizing radiation, and we saw also that the survival curves are reacting to this exposure of ionizing radiation. And if we compare these results of desiccation and survival, we saw that there is no general correlation between desiccation and radiation tolerance, as it was supposed on the basis of Deinococcus radiodurans, which was the one organism where first it was claimed that the desiccation and radiation tolerance is correlated. And we have some more archaeal examples where we do not find this correlation. And one example is Ignicoccus hospitalis, which is highly tolerant to ionizing radiation, but not to desiccation at all. The second protection mechanism can be the protection mechanisms against osmotic stress. Just to inform you, there are different levels of organisms who are tolerant to different salt concentrations. We have, for example, halotolerant organisms, we have halophilic organisms and extreme halophilic organisms. And for example, one halotolerant organism is Staphylococcus aureus, and one extreme halophilic organism is Halobacterium salinarum. And where are these organisms living? They are living in, for example, these areas where salt is produced, sea salt, and also in areas like the Dead Sea. And there we find these extreme halophilic organisms like Halobacterium salinarum. And also you see here, we have again this pinkish color in the colony. However, there are very strange or super interesting shapes of organisms in these very salty environments. This is Haloquadratum valbusi, and the cell, this is really the cell which had this unique shaped form of a quadrat. And yeah, I think this is from a microbiology point of view, very interesting and really nearly funny. So the question was, are these protection mechanisms also helping during desiccation? Or which protection mechanisms? We have, again, as I said, the pigments in the cell wall, which are helping against UV radiation and the reactive oxygen species. In halophilic or even superhalophilic organisms, we find enzyme stabilization by compatible solutes. I will talk about this later. We have proteins with highly negative surface charges. We have, again, different DNA repair capabilities, and these organisms are very metabolic diverse. So we've asked us, are, for example, cells which were exposed to a slight osmotic stress more tolerant to desiccation? And here we have the example of Archaeoglobus fulgidus and Hydrogenothermus marinus, an Archaea and a bacteria. And Archaeoglobus is normally growing at 1.8% sodium chloride. So we doubled the concentration and dried the cells. And what we saw, that the survival rate was changing. The cells survived better if the cells were grown or stressed before the desiccation treatment with a little bit more salt. And the same we saw with Hydrogenothermus marinus. So this organism is normally growing at a sodium chloride concentration of 2.0. So if we double the sodium chloride in the media before the desiccation treatment, we saw that the survival was higher than under optimal growth conditions. And in both cases, we saw that the cells were producing compatible solutes. And we assumed that these compatible solutes, which helps the cells during this mild osmotic stress, also helps the cells during desiccation. But we saw also that there are more archaeal examples where we do not find this correlation between mild osmotic stress and desiccation tolerance. Here again, we have Ignicoccus hospitalis, also Thermoproteus and Pyrococcus. And for Thermoproteus and Pyrococcus, it is also known that they can produce compatible solutes under mild osmotic stress, but there was no desiccation tolerance visible. So there is no general correlation between desiccation and salt tolerance. Then we asked the question, maybe this desiccation tolerance is correlated with the phylogenetic position in the tree of life. And we tested a broad variety of archaea and bacteria against desiccation in the point of view of desiccation tolerance. And we saw that many organisms from these archaea and bacteria are desiccation tolerant. But we have an influence of the pH during storage. For example, this was true for the sulfolobales strains. We had an influence of oxygen during storage. This was mainly for the methanogenic archaea. We had an influence of the temperature during storage. This was true for Ignicoccus. At cold temperatures, in the absence of oxygen, Ignicoccus can survive desiccation. And we saw that if you have a look at the cultivation temperatures, though we have a huge range between 37 and 100 degrees, there was no correlation with the growth temperature. We had no correlation with the origin or habitat. So we cannot say all organisms which are coming from terrestrial extreme environments, for example, like the sulfolobales, are tolerant, or all methanogens are tolerant. So there we didn't found a correlation with origin or habitat. We also did not found a correlation with metabolism, as I already said, for example, that all methanogens are desiccation tolerant. And this is maybe the key information of the whole talk. Desiccation tolerance of vegetative cells, so we are not talking here about spores, is a highly species-specific and strain-specific property. So it can be that, for example, one organism like Methanosarcina bakeri is desiccation tolerant, and Methanosarcina matsui is not desiccation tolerant. But desiccation tolerant microorganisms can not only found in natural habitats, we find them also in artificial dry areas, for example, in clean rooms or even on the ISS. The common characteristics of these areas is the dry condition. We have air filtration and strict cleaning procedures. And we had we were part of one project called Extremophiles, where the microbiome was analyzed within the ISS. And we are doing regularly some determination of the load and distribution of microorganisms in the clean rooms and where spacecraft are assembled. We do this together with ESA, and this is all under the umbrella of planetary protection. And coming back to the results of the Extremophiles project, and there we had 3 campaigns or times where surface samples with wipes were taken, and the results were compared with clean rooms on Earth. And we saw, or the results are that we find archaeal and bacterial sequences. We have differences in the composition of the genera between the different sampling campaigns, and the ISS microbiome is subjected to a change over time. So that means that every single spot, even taken at the same area, are different, and during the different sampling campaigns. And we also saw that the locations where the samples were taken, for example, at the spot area at the treadmill, is very indicative for the samples. So that means we find there organisms which are only found in this area. In general, we found a core microbiome of 52 microbial genera, and these core microbiome, or all the samples, are similar to other closed environments like the clean rooms or some rooms in hospitals. Here we see also from the Extremophiles project a tree of isolates which were cultivated from the samples we got back. And these samples were treated before they were cultivated with UV and X-ray. So we see that there are organisms on the ISS in this dry environment which are able to survive a treatment with ionizing radiation. And the question is, are they also desiccation tolerant? And this we should investigate in the future more in detail. But dry conditions in combination with radiation and salts, we do not find only on Earth. We find these conditions also on Mars. How is the situation on Mars today? So we have no liquid water. We have the regolith, the soil on Mars. We have oxidizing compounds like perchlorates and hydrogen peroxide. We have high salt concentrations and low nutrient content. Additionally, we have the atmosphere, the low pressure of 7 hectopascal. We have anoxic conditions. There is nearly no oxygen and mainly CO2. And we have low temperatures, on average minus 65 degrees. And we have different ionizing radiation environments on the surface on Mars than on Earth because Mars do not have a magnetic field, and we have higher UV radiation on the surface of Mars because we have no absorbing or protecting ozone layer on Mars. So to sum it up, we have their desiccation conditions, we have their radiation, perchlorates, salt, and regolith. And we asked, can, for example, anaerobic microorganisms, anaerobic because there is no oxygen on Mars, can these anaerobic microorganisms survive simulated Martian conditions, including the desiccation and the radiation environment? And for this, we had several years ago a project called MACE, Mars Analogs for Space Exploration. And to make the long story short, we went to different Mars analog environments, got some samples. We had over 1,000 different inoculations, hundreds of enrichments. And finally, we got 31 facultative anaerobic isolates. And here we have these two organisms I already named before, Butteoxella and Yersinia. And at the recent time, we also included Salinisphaera in our studies. And with these model organisms, we tested the ability of them to survive after Mars relevant stress factors. We tested these stress factors in single and in combination, and we always started with anoxic desiccation. We included radiation, vacuum temperature shifts, oxidizing compounds, and the lack of nutrients, and many more. How did we do this? So first of all, we cultivated our anaerobic microorganisms in these serum bottles. We dried them on glass plates or glass discs, also in the presence of artificial Martian regolith. So there is a company, Exolith, in the US, where you can buy this artificial Martian regolith. And then we expose the cells to vacuum or to Martian atmosphere, to ionizing radiation, to UV, and then we determined the survival rate or did some microscopy. And here are the results here is a description of the cells before. We have Butteoxella, Salinisphaera, and Yersinia. All three organisms are bacteria. Salinisphaera and Yersinia are belonging to the gamma proteobacteria. All three are methophilic organisms. They are facultative anaerobic, and Butteoxella is coming from the Iselinger Mühlbach, a sulfidic spring nearby Regensburg in Germany. We had Salinisphaera shabanensis from a deep sea brine pool in the Red Sea, the Shaban Deep, and we were working with Yersinia intermedia from Lake Granavatn in Iceland. And here are the results for Butteoxella. Butteoxella is able to survive desiccation, radiation, the exposure to regolith, and the exposure to perchlorates. That means, for example, if Butteoxella is dried without the addition of the Martian regolith, the survival is going down faster and lower than in the presence of the regolith. The same we saw that when the cells were irradiated in the presence of the regolith and in the combination of desiccation and irradiation treatment. Here we used polychromatic UV as we would expect it on the surface on Mars. So we saw some protection due to the added Mars analog material. And all these experiments of the combination of desiccation and radiation were done in the T-Rex box. It's a transport and exposure box where we can put in the dried cells and irradiate them under anoxic conditions. And Butteoxella is also able to grow in the presence of perchlorates. However, we find a change of morphology. So these are the cells without any perchlorates, and these are the cells if a Martian concentration of sodium perchlorate is in the media, and we get these long chains of cells. Why are we doing this? Butteoxella, MACE IM9, is on the passenger list for a 3-month passive exposure experiment on the outside of the ISS called MAXEM. Here are some results for Salinisphaera shabanensis. Salinisphaera shabanensis survived for 5 months under Martian conditions. So we had Martian atmosphere and the low pressure, including a stratospheric balloon flight. So here you can see where this flight was done. And here a picture from the stratosphere and the T-Rex box in the stratosphere, and the lid was opened when the balloon was in the stratosphere and then was closed again, and thereby we stopped the exposure to the UV. And we saw that the UV shielded cells, so we had 2 layers of cells, one directly exposed on the surface and one below, that the UV shielded cell survived this complete mission scenario, including the balloon flight, and the UV exposed cells, there was less survival because they were exposed to UV during the balloon flight. And also Salinisphaera shabanensis is on the passenger list for the space exposure experiment. And then we had the question, how is the desiccation tolerance in an artificial community? So we mixed Butteoxella and Salinisphaera and dried them together. And we saw that there is a slightly better survival of the 2 strains together than separately. So we see that here we have the mixed strains, and here is the survival of the single strains, that we have a little bit higher survival if the cells were dried in this mixture. And this was even true if we were mixing 3 different strains, Butteoxella, Salinisphaera, and Deinococcus geothermalis. Deinococcus geothermalis is also very desiccation tolerant, which you can see that the survival rate is not dropping that fast. And also here we see that the survival together is a little bit better than separate. Here are some results for Yersinia intermedia, maize LG1 from the Lake Grenavatn in Iceland. And we saw that these organisms coming from a liquid environment of a lake is able to survive without any addition long-term desiccation, up to 200 days. And this is very special because other Yersinia strains, like the bad cousin Yersinia pestis, is not desiccation tolerant at all. We saw for Yersinia also that there is an influence of pressure and atmosphere during desiccation. And where this arrow is the marking, we see that it's the influence of the pressure, and we have even a bigger influence if we are applying the Martian atmosphere. And we saw for Yersinia that the perchlorates are also influencing the desiccation tolerance. So here we have the survival after desiccation of 24 hours in the presence of perchlorates in Martian relevant conditions. We saw that the survival rate was much lower than without the perchlorates. And with this, I want to sum up my presentation. I think extremophilic microorganisms have extended our knowledge of life at its physical and chemical limits. Probably there are much more representatives and extremophilic microorganisms on Earth than we expected it a few years ago. So they are really everywhere. And desiccation tolerance of vegetative cells is a widespread phenomenon and can be influenced by several biological, for example, this mixing experiment, chemical, for example, the addition of perchlorates, and physical parameter. But just to repeat it once again, because I think this is the main thing you should keep in mind, desiccation tolerance is a highly species-specific and strain-specific property. And with this, I want to thank you for your attention. If you have any questions, recommendations, advices, just let me know. If we do not have the time now to answer the questions, just write me an email and I would be happy to answer you now or later on via email.